Well, good evening, everyone. Thanks for coming, and thanks to the Simons Foundation for the invitation to deliver this presidential lecture. It's a real honor for me. Also a pleasure to have my family here with me as well. My wife and two kids are here. My kids have been forced to attend. This morning my dad said, my son said, "Dad, are you going to talk about that stuff you always talk about? And if so, can I skip?" But maybe you'll learn a new thing or two today. It's also fitting that today is Valentine's Day, and I bought my wife some flowers today. And as she saw the flowers, the first thing she did, I'll also say there was a little bit of force and coercion there. But after she received the flowers, we embraced. There was a hug, right? And why do we do that? It's because we crave this social touch, right? It gives us connectedness and warmth, and that's what we're going to talk about today. It's with my lab studies, trying to think about what are the genes, the molecules, the pathways that allow us to detect these sensory cues in the skin to mount appropriate behavioral responses. So what you see here, maybe I'll use this. Our skin is actually our largest organ, and it's constantly bombarded with myriad sensory stimuli, things that are hot or cold or itchy or pleasurable or painful. And we need to, again, relay that information to a central nervous system, our brain. And I think all of you would appreciate that. There are a lot of neurons in our brain, but we also have another nervous system in our peripheral nervous system. There are sensory neurons there. And these two systems are talking. They're synaptically connected, our peripheral and central nervous system. So as you'll see today, we're quite interested in making the connection between our outside world and inside world for social touch. And I hope to be able to convince you that studying these things in a mouse and a naked mole rat can teach us some fundamental neurobiology. All right, so as I tried to allude to, and it doesn't take any time to convince you all that social touch is important. It's the foundation of our social relationships. And I think the COVID pandemic is a great example of that. We all clearly suffered a lot of deficits, and one of which probably stemmed from the fact that we were socially isolated and we weren't getting the touch from our friends and loved ones. That's just foundational to our daily lives. But despite the fact that this is so critical, again, the genes and neural circuits and pathways that drive this social touch remain poorly defined. And this is why we're hard at work in my lab to try to learn more about these things. All right, so the so-called somatosensory system, soma just meaning body, so bodily sensations. Here's a rough neuroanatomical sort of wiring diagram of how this system works. So if there's some sort of stimulus in the periphery, there is detected by these first-order sensory neurons, that relay information to the spinal cord, and then ultimately to interconnected networks throughout our brain. And we're interested in probing the system across different nodes in this relay, from the sensory neuron, spinal cord, and the brain. Mainly today, I'll tell you about work in the sensory neurons and networks across the brain. So, I stand here today as a representative of an entire field, I would say, and somatosensory neurobiology and research. And it's been a great few years for colleagues of ours here in the entire field with this recognition. So the Nobel Prize, if people don't know, was awarded a few years ago to friends and colleagues of ours, David Julius and Artem Patapushian, for really groundbreaking discoveries. So Artem discovered, in his lab, discovered a receptor protein called piezo2, which is expressed in those sensory neurons, and it's really important for touch and mechanical sensation. David Julius discovered a different receptor protein called TRPV1, which is important for pain and temperature, noxious sensations. So, these, again, I think we all in the field can stand a little bit taller with this recognition and new sort of levels of spotlight on our field. But I would argue that, it's still early days, okay? Just because a piezo2 receptor or a TRPV1 receptor is activated in the periphery, that still doesn't tell us how we perceive these things, how it interacts with physiology across the rest of the body and brain. So there's still lots of open and important questions that we all are thinking about together in the field. here was a recent meeting. I actually co-organized this Keystone meeting on touch and somatosensation. It was a lot of fun, some of the leaders in the field, and we got to discuss big open questions that we're all still quite interested in. And again, I just want to convey the point that I stand in front of you today as a representative of an entire field. And I'll just show you some of the things that we're up to in my lab. All right, so moving closer to my own lab, just to give credit where credit is due, as I talk about our work in the mouse first, this work was led by two of my first two PhD students, Leah and Melanie. They've since graduated and off to great things, and two former technicians, Bella and Will, who've recently come back to Columbia to join the lab as PhD students, and I'll be able to share today some of their new work as well. All right, so the neurons that I'll begin to tell you about are these neurons called C-low threshold mechanoreceptors. So I don't have time to tell you about all the sensory neurons that as a field we've discovered that exist in our skin. And we can characterize them based off gene expression, neuroanatomy, physiology, function, etc. But one class of neurons that seems really important for social touch are these neurons called C-low threshold mechanoreceptors, okay? Every mammal that's been studied, these neurons appear to be very important for stroking touch. They're probably not the only class, but it's one that the field has kind of keyed in on. So this particular paper, they were first identified in cats in the late 1930s in this paper. This is not a cat from that paper. This is my own cat, Bella. Anytime there's an excuse to show your own cat. But what the investigators did in this paper was stroke a cat across the skin surface, and they found these neurons that fired to the stroking touch. But the neurons did not fire when they gave something painful to the skin, showing that there are dedicated nerve cells in our skin that mediate the stroking touch. Okay? And since this paper, almost every mammal that's been studied appears to have an equivalent population of these neurons. They were first identified in humans in this paper in 1990. And in humans, you can talk to people, so-called psychophysics, and combine with electrophysiology. So in this study, they took a person and they stroked this cotton wool slowly across the person's skin and recorded the sensory neurons, and there are a lot of neurons that fire towards the stroking touch. And you ask the person, how does this feel? They say, this feels really good. Okay? But if you gave them something painful or a fast stroke, the neurons didn't fire. So it seems to be dedicated neurons that mediate these social cues, which the same has been seen in most mammals. So now closer to our own studies. Another background slide before I get into our work. So this is a particular paper from David Anderson's group at Caltech, and came out some years ago. Now, I read this paper as a postdoc and got super excited because in the mouse, the Anderson lab found a putative population that bear similarities to these neurons I've been telling you about that seem to be these gentle touch detecting neurons. So this is in the mouse, and this particular gene called MRGPRB4, it's a G protein coupled receptor, but it's only expressed in this small population of sensory neurons in the skin. And here they measured neural activity in the mouse, and what you can see, similar to what I showed you in a cat and humans, if you just stroke the side of a mouse, these neurons fire, okay? But they didn't fire if the animal was pinched or given another noxious stimulation. So all consistent with these being these gentle touch detective neurons. So one of the things that we were interested in studying when I opened my lab, two things I would say. One, we wanted to ask, are these particular neurons important for any natural behaviors if they're these gentle touch detective neurons? And I think more importantly, if they mediate this social touch, and this is rewarding to the animal, how does it engage with reward perception and reward circuits in the brain? And we wanted to make that connection. So as a starting point for these studies, we began using this technique called optogenetics. And for all the neuroscientists in the audience, you can just cover your ears on this slide. But for everyone else, just to follow, optogenetics is a really important, revolutionary technology in neuroscience for the past 15 years. It's allowed us to interrogate neural circuits at a really high resolution. And it makes use of this blue light sensitive ion channel or protein called channel rhodopsin. And neurons are electrically excitable cells. So if you can put this opsin protein on the surface of your neuron and hit it with blue light, the channel will open, allow positive ions to flood into the cell. This activates the neuron. So if you can put this channel-rhodopsin protein on the surface of your favorite neuron, you can activate it in vivo in a behaving animal, okay? And optogenetics helps us as a field. One problem of neuroscience is the problem of scale. There's just so many neurons, millions of neurons in our head. How do you activate one cell without activating its neighbor? It's nearly impossible. But with optogenetics, it allows you to kind of, activate individual trees within a forest of neurons. But most neuroscientists who use optogenetics will activate neurons in the brain. We reason that we could put this protein into neurons in the skin using genetic targeting, and perhaps this could mimic touch, social touch in animals. So this was our point of departure in this project, okay? So we used the mouse, and one reason we used the mouse is because we can do these sorts of genetic, wizardry. And so we put this channel-rhodopsin protein into these B4 neurons in the skin, and there's a green fluorescent tag so we can visualize where these neurons are. So here's a cross-section through a mouse skin. You see this is actually a hair follicle here, and these neurons are in the hairy skin. These green lines are sensory neurons, and they all express this blue light sensitive channel. So the reason why the experiment works that I'll show you on the next slide is because the top here is the surface of the animal skin, and if we shine the blue light directly to the skin, we can activate these neurons. So this is what we did, and this is what we saw, which is quite interesting, I think. So here in this first video, this is a wild type, a control female, and this video, please. Come on. Oh, maybe I need to turn. Wonder if I need to turn this pointer off.
Take the pointer off, yeah.
Okay. What should I make it? Okay, there we go. All right. So here's this first video, pulsing blue light to this animal's shaven back. The animal recognizes there's an experimenter in the room, but doesn't really alter its behavior in any meaningful way. But look at this next video here. Hopefully you can appreciate that this animal is like lowering its back and adopting this posture as if it's been stroked. Maybe back to the cat example, sometimes if we stroke our cat, it kind of like makes this back lowering sort of posture. I won't get into all the data today, but we've done very similar experiments with other lines where we can induce pain or itch. The animals don't adopt that posture. So it seems like the animal is responding as if it's been stroked. Okay. So we can quantify this sort of back lowering phenotype that we see in experimental but not control mice, and we see a nice statistical separation between our experimental versus control. We don't see the same thing when we activate a related population of sensory neurons in the skin. So next we designed this experiment, which is common in behavioral neuroscience. So we put a slight twist on it because we're doing this peripheral skin optogenetics, where we do these so-called conditioning experiments because the animals can't talk to us. That's one limitation in using the mouse or any animal species for studies. So we have to try to make our best approximation based off their behavioral neuroactivity. But anyway, what we do here is just ask the animal, to essentially vote with their feet. Where do you like to spend your time? So we have different conditioning stimuli on each side of these chambers. And whenever the animal is on this side, they get this blue laser light, which turns on that blue light protein, sensitive protein. And here this is green light, which the animals can also see, but it won't activate that protein. We do this for 5 days, and then on the 6th day, we remove the lasers. And this asks where do animals prefer to spend most of their time. And indeed, these animals spend more time returning to this environment where they receive this blue laser stimulation. All consistent with the animals finding this is a good proxy, we believe, for the animals perceiving this as rewarding, something that they like. And we don't see this with the other population. Here's a study that came out from a group in the University of Toronto at the end of last year. And it's cool to see, others are already, sort of following up on some of our discoveries. So they used the same mouse line and a very related technology activating the neurons with blue light in one chamber versus no stimulation here. And very similar to what we see, they see the animals do prefer activation of these neurons. Okay, so I think it's always good in science when you have outside investigators independently arrive at very similar conclusions, and consistent with animals finding this stimulation on their skin as rewarding. So next we wanted to look at behavior. Well, what are some natural behaviors that may rely upon these neurons? One that we began to study is mating, which is a rewarding behavior in mammals that requires lots of social touch. And what we did, won't get into the details, but instead of activating these neurons like I told you before, here we genetically ablated the neurons and looked what happens when the animals engage in mating behavior. And this is what we see. So we're looking at female sexual receptivity, responsiveness to a male. And we see this receptivity goes up over time, over subsequent weeks, but we see when these touch neurons are ablated, sexual receptivity in this mating assay is greatly diminished by the second and third week, okay? What are these females doing when they're no longer sexually responsive? They actually become quite aggressive towards the male attempts to mount, as shown here. There's a lot of fighting off the male attempts to mount, which is something that you almost never see in a female mouse that is primed to be in a state of behavioral estrus. So what we can say from these experiments are that indeed these neurons, at least in the mouse, are absolutely required, critical for sexual receptivity in the female. And what we think is that they're not just required for the sense of touch per se, because one of the things maybe I haven't mentioned to this point, this is a relatively small population of sensory neurons, only maybe 5%. So these animals have a lot of touch neurons, so it's not that they can't sense that they're being touched, but we think is that they're really critical for this positive reward network, such that if you don't have this signal in the skin, then you won't get this reward signal to touch in the brain. So if that's true, how can we kind of prove some of these things? So we thought we need to turn to the brain and connect these studies. So I'll show you a few sets of experiments to get at that. So one is this, where we do this technique which has become increasingly common over the past 5 years in neuroscience, this technique called pyrophotometry, and we measure dopamine release in the nucleus accumbens, so a well-known reward center in the brain. And we combine this with that skin optogenetics I've been telling you with. So we have this high affinity dopamine sensor. When dopamine binds this sensor, it emits photons of light that we can detect with a photodetector and read on our computers. So when we do these experiments, we see that indeed shining light to the back skin of these animals where these neurons reside gives us a nice dopamine release, our experimental but not control condition. When we shine the light to the back skin, or even in the genitalia where these neurons also reside and where this organ is touched clearly during mating, we see this nice dopamine release, but not to our control light. So these experiments tell us that indeed turning on these neurons in the skin is sufficient to engage this canonical reward circuitry. So it's an independent readout beyond the behavior, but in reading brain states. So what about necessity when we ablate the neurons? So we went back to our dopamine recordings, but in the context of mating. And what you can see here is that if we align these dopamine traces to the male mount onset during mating, we see a nice dopamine increase and control females, but in the touch neuron females, this is lost, and that's quantified there. So we think these recording experiments do provide a compelling explanation as to why the females are no longer sexually receptive, because they've lost this reward signature. So we published these results in a journal about 1 year ago at this point. And I think, and I kind of went over a lot of the main findings there, but there's a paper if people want more details. And I think this is, we're excited about this paper. You know, it shows this sort of skin brain or brain body physiology and connectivity, and it's opened up many more questions for my lab. Now, what I'll do on the rest of my time is tell you about all unpublished and ongoing studies in the lab. So shortly after we published our paper, another group wrote sort of a summary or news and views style article on our work. I thought this was a really nice synopsis of our work. One of the things that I didn't have time to talk about, but you can see that the authors depicted here based off our studies. You know, you may ask yourself, well, how do you even get from the skin to the brain's reward center? Well, we showed in the paper, I didn't have time to go through the data, using a series of physiology and viral tracing approaches that about 3 to 4 cells or synapses can get you from these neurons in the skin to these neurons in the spinal cord, from there, an area in the brain called the parabrachial nucleus, to the VTA neck. Okay? We think this is probably one of many pathways that you may engage the reward circuits from the skin. One of the things that the authors, as they concluded this review of our paper, they said, you know, this work is nice, but the authors, we only looked at sort of one circuit, the VTA to neck. But if you think about social touch, it must engage more of the brain. The animal has to localize, where is this touch coming from? Touch doesn't feel the same depending upon where it's delivered, who it's delivered from, the context, internal states. There are all sorts of things that go into this perception. And it would be nice if the authors or people interested began to take a holistic approach. As we read that, you know, we couldn't agree more. So we're starting to, this is one approach we're taking to kind of go after this. And this is led by a PhD student in my lab, Bella, who was a former technician on the earlier work. And this has been done in collaboration with a colleague of ours, Itamar Khan at Columbia, the Zuckerman Institute, and a graduate student in Itamar's lab, Amir, has helped us immensely. So what Bella wants to do is to generate a brain-wide representation of social touch using the technologies that I've told you about from my lab in collaboration with Itamar's lab. And what Itamar's lab he's spent like the last 10 years or so trying to pioneer fMRI in the mouse, awake fMRI in the mouse. So we've been putting our heads together. And so let me show you a little bit of where this is. So fMRI, for people who may not know, it's an important brain imaging technology, but mainly used in humans, almost rarely used in rodents. So this is sort of a newish paradigm. So we have this 9.4 Tesla in this big room and big scanner for a tiny mouse. And here's the mouse as it's about to go through the brain scanner to get its brain scanned. Hopefully you can see its face there as it's about to go through. The animals get habituated and used to this environment. Each recording is about 45 minutes or so. And importantly, Bella and Amir, we have lasers that are going into the scanner, okay? So we can deliver that blue laser light to the animal's back skin. And the reason we like fMRI here is because we can get the whole brain activity in an unbiased fashion, okay? As we're pulsing that blue light to their back, which we've already shown the animals seem to prefer. So these are early days, but so far it's starting to look like it works. So you can see here, and we have relatively nice temporal resolution as well. So here we're pulsing the blue light. And as we pulse the blue light, we see areas activate like the nucleus accumbens, which is a bit of a sanity check that's consistent with our previous work. But we see the hypothalamus, which is an area important for social connections. The cingulate cortex, an area very important for reward and emotion. Somatosensory cortex, but the trunk and not the mouth, okay? These are early days, but it looks like the technique is working, and I think this will be quite powerful for us to, perform these skin-brain sort of experiments. And we want to be able to create sort of a hotspot of reward across the entire body. Remember, with the peripheral optogenetics, we can just shine spots of light. So we hypothesize that we shine spots of light in areas that are social, like the back or the face, we'll get a different representation versus shining light under the arms, for example, in non-rewarding areas. Or, what happens if you change the internal state of the animal? The animal is stressed or hungry or fearful. What does this representation look like? These are all questions that we're quite excited to go after. Another new set of data that we're excited about, a new-ish postdoc in the lab, Sasha Fulton. She comes from Ian Mays' lab at Mount Sinai, so a lot of sequencing omics approaches in the brain. And Sasha was struck by the idea that when we ablate these touch neurons, the animals have these deficits in, rewarding behavior. So we decided to take a gene expression-based approach. Okay, so RNA sequencing in the nucleus accumbens when we've ablated these touch neurons. This is a reward center in the brain that we've been imaging from, but now we're looking at genes. And this is a silly configuration, but this is all the rage in the single-cell omics. This is a UMAP projection of a known genetically defined cell type. So this is just a sanity check that we can reveal the known cell types in the accumbens. But what's more important, what I want to draw you to is this. if we go in and look at these clusters and look at individual genes, we see something quite interesting. So this is the D2 dopamine receptor. We see it's greatly downregulated when the touch neurons are ablated. Okay? So this means that just ablating a receptor in the skin that's important for touch, which I showed you before, can lead to downregulation of a receptor in the brain important for reward. I think that's a quite striking result. And it's not just a dopamine receptor, but if we look at dopamine response genes, so we put this list of genes together based off others' work, and we see an entire network of dopamine responsive genes is downregulated when these touch neurons are ablated. Okay? So this is all consistent. I see there's a question, but I think they want me to take questions at the end. I can take one now? Okay, let me just finish this slide. So you know, together, these data do support our findings that these touch neurons are really important for reward, and when they're offline, you know, reward circuitry is greatly diminished and altered. Yep. Happy to take.
Yeah, thanks. No, I'm enjoying it. I was going to wait to the end, but I wanted to ask, since it's coming back up and it might affect how we think about this. The ablation of these neurons, is that through some technique where it's acute, like they did not develop, like they develop normally and then they're ablated, or did they develop without these neurons?
This is a critical question that I glossed over. So the question is, are these ablations occurring right before we do the experiments from behavior or imaging, or did animals grow up like this? So this ablation occurs at early life. So this gene comes on about postnatal day 5, and it's just a straight Krelux sort of cross. So the animals grow up without this circuit, and I think that's why we see something so striking like this. If it was just an acute ablation, I think we wouldn't expect to see this, but this is basically taking an adult animal that's grown up without this reward touch sort of network, and I think this is why we see quite profound effects here. It's a great question. All right, so the last bit of data I'll show you is this before we move over to naked mole rat. So this is part of a collaboration with Catherine Dulock's lab at Harvard. This work here was led by the Dulock lab, but we played a small but important role, I would say. So the Dulock lab has been using this paradigm, which is quite interesting. So they take mice that have been hanging out together, cage mates, and separate them from each other, either isolated 1 day, 3 days, or 5 days. Then they bring them back together to hang out. And what they see is this. Depending upon how many days the animal has been isolated from its friends, when you come back together, they actually spend more time wanting to engage with each other, as shown here. And they notice when the animals come back together, they seem to be huddling and doing a lot of social touch. And this is why Catherine got in touch with us based off some of our work. So this paper is really about cell types in the brain and the hypothalamus that seem to be important for detecting isolation and promoting reunion. But we also looked with our touch neurons, and we see this. So when these touch neurons are ablated, normally animals after 3 days will have this drive to get back together with their friends, but this is greatly diminished without these touch neurons that we've been talking about. It's not down to 0, suggesting that there are other neurons probably important for social touch, but this deficit does give us yet another behavioral context that involves social touch, where these neurons seem quite important. All right, so to summarize this first part of the talk, and think about ongoing questions, you know, we're quite interested in the cells and circuits that promote social touch. Again, this is kind of one that we outlined in that paper, but there could be other pathways, and we're really interested in interrogating this circuit at high resolution from the level of genes to circuits. I mentioned this briefly, but you know, how does internal state alter the brain's representation of touch and social touch? Everything I presented today, you could argue, is overly simplistic, as if you just hit a button, hit a light switch, and then you get reward. But we all know it doesn't work that way, right? Reward that's delivered in the comfort of your own home, or touch delivered to you from a friend or loved one in the comfort of your own home is different than if someone touches you in the 1 train on your way to hear me talk, right? But it could be the same touch, but you interpret it quite differently, right? How does the brain prioritize how you should respond? I think these are questions that we can get at with these technologies. And what are other behaviors that require this social touch pathway? I just focus on mating today, but we're interested in studying other behaviors. For example, a new postdoc in the lab, Brianna, is quite interested in lactation and mother-pup interactions, because these neurons are everywhere where there's hairy skin in the animal and we see a lot of innervation of the mammary tissue with these neurons. So we're investigating that as we speak. All right, so let me move from mice to naked mole rats. And hopefully it'll be clear to you why we're interested in naked mole rats and some of the discoveries we're starting to make already. And here's Team Naked Mole Rat, the brave folks who are trekking out in this direction in my lab, and someone who hasn't even started yet, who's here in the audience, but will be joining Team Naked Mole Rat. All right, so here is one of our lovely critters. So this animal is an animal walking through a tunnel in our interconnected colony system at Columbia. You know, if you squint your eyes and tilt your head, you know, not so ugly. So there's the naked mole rat. So they have this eusocial organization. So just like bees and ants, there's a king. So this is a queen here. The queen is the most dominant. There's a queen, there's a breeding male, and a bunch of subservient workers. And they live in these underground colonies in Kenya. And within their colony, you can have dozens or even hundreds of animals all living together, stacked on top of each other in these underground burrows. And clearly there's a lot of social touch that they engage in. And because they live underground, there are a lot of unique adaptations that these animals have. For one, they're essentially blind. They have these vestigial eyes, but you know they live in complete darkness. So vision has been lost in these animals. They don't seem to be able to hear so well. You see that their ears are basically lost. The olfactory epithelium is atrophied. They don't seem to be able to smell so much, but it seems like touch is heightened in these animals. So they have these whisker-like hairs that you see in the face, but they tile the entire body. And I'll show you in a few slides that they're very exquisitely tuned. So for a lab like ours that's interested in social behavior, social touch, and these skin-brain connections, these animals present an extreme model to study that. And I think by studying animals that have these extreme adaptations, perhaps we can learn some fundamental biological principles. All right, so as I mentioned, they have this extremely cooperative lifestyle, and it's been suggested, but not, I don't think, firmly proven that they have this caste-like organization of behaviors. As you can see in this cartoon, some animals are digging, the queen is over here in a nest, some animals are foraging for food. It seems like they organize behaviors in a caste-like fashion for the ultimate cooperation in their society. So, right, and as I kind of got at, this paper 20 years ago showed that their touch system appears very exquisite. So those body hairs on their body, if you just deflect a single hair, they will orient their entire body towards that deflection of just a single hair. So that their touch system is highly tuned and exquisite. And this one of the only papers that have ever done brain recordings in these animals, in a anesthetized animal. They kind of did a so-called somatotopic map. And what you can basically see, if you look at a standard mammal, a lab rat, here's somatosensory cortex, here's visual cortex. Because these animals are blind, they basically lost visual cortex, and somatosensory cortex has appeared to have encroached upon it. So somatosensory cortex in these animals is 3 times expanded compared to other mammals. Again, fitting with this idea that somatosensation is their prominent source of sensory inputs. So a great model, I think, for us. So let me show you some of the things we're starting to find and discover in these animals. So one of the things as a point of departure, we just wanted to ask about their behavior and whether they do have caste-like organization of behavior. So to do this, we deploy some modern tools in systems neuroscience. Computational neuroethology is a fancy name for the field. And the neuroscientists in the audience will be aware, but maybe for everyone else, I think the field of neuroscience is definitely encountering a renaissance, and a lot of using AI, machine learning, to automatically measure and quantify behavior. So here we've just been using some technologies from friends and colleagues in the field. So we use this one program called Sleep to use deep neural networks to track animals. So we can track one animal, 5, 20, whatever, simultaneously, as you can see here. Hopefully you can see labels on these animals, so we can keep track of animals as they're engaging. And then we use this program called, so MoSeq was developed maybe 8 years ago from Bob Datta's group, and the newest version is called Keypoint MoSeq, where you can feed in this keypoint tracking data to MoSeq for unbiased behavioral discovery. And then we get all sorts of behaviors. Here's an example of a behavior that MoSeq scores, and this is a digging behavior, for example. I won't get into all the data, but I'll just summarize it quickly in that we see evidence of behavior organized in a caste-like structure. We do see like the workers only dig, for example, and the queens never dig, but the queen does this fast darting behavior, which is probably consistent with her patrol of the colony. So using these unbiased machine learning tools, we do see evidence of behaviors that are organized by caste. So one of the things we've done is look at these interactions in mole rats. So as you can see here, again, they live underground and they're building these burrows and tunnels, and the tunnels, they frequently come encounter with one another. And when they come encounter with one another, they do this behavior I'll show you here. And let me just play this high-speed video. So what's occurring is happening in a few seconds. And what you'll see is that the dominant, as they come to cross each other in these tunnels, so this is our experimental setup in their system, the dominant animal will crawl on top and a subordinate animal will go underneath. All right, so they come together, they touch faces, and then they make a decision. Dominant animal crawls on top, subordinate animal goes below, okay? They're doing this constantly in their native environment. So this is like a hardwired behavior for these animals. But one thing that really struck us was this face touch, okay? And I'm gonna be coming back to this point in the next few slides. Because at this point, we've taken about 20 animals, and we've done these tests, these trials. We gave every of these 20 animals every pairwise combination and 10 trials each, so about 2,000 trials total. So in all 2,000 trials, before they make the decision about who's dominant or who's submissive, they touch faces first. All right, so when we do these things, we can get a rank order of these 20 animals and of these 2,000 trials, and we see it's, I mentioned this deterministic and relative. Deterministic meaning that you can see this here. If there's a larger rank differential amongst the animals, if we pair number 3 with number 15, we can, 3 will win at a pretty high rate. But it's relative in that, if animals are closer matched, then it's more random. So you're not absolutely dominant. You can just be more dominant than depending upon where you are in this chart. But the interesting thing here is that it's quite stable. They all seem to know their place, and they all seem to know one another. And again, they face touch. So could face touch, this is a question I'll raise today, like could face touch be a part of their recognition system? We don't know, but we are hypothesizing this. More about this face touch. We notice too that when they do these encounters and they always face touch, if we have animals that are more disparately ranked, the high versus low, this encounter is very fast, 40 milliseconds. They always face touch, but it's fast, and then they go towards the behavior. But if animals are a little more closer in rank, they take about 10 times slower. They're doing more of this face touch. We think perhaps they're trying to gather more cues, like who the hell are you? And once they know their place, maybe they engage in this behavior. That's a speculation that we need to prove. But more about this face touch. So instead of using this tube test, we just wanted to look in a more open, naturalistic environment where we just had 2 animals freely engaging. And we used sleep tracking. We wrote our own code as well to keep track of these face interactions. And what we saw, I think, is striking, that over a 10-minute window, the animals do this face touch hundreds of times. Right? Why do they need to keep touching faces so many times? We don't know. And again, if we extrapolate to an entire day, they're probably doing this thousands of times. Okay? If we put two farm mole rats together, as I'll show you in a second, so farm mole rats are quite xenophobic, just like many people. They don't like foreigners, and they'll fight, okay, sometimes to the death. How do they recognize a foreigner is unclear, but they're able to. But one thing we see is that when we've gotten different mole rats from other colleagues around the world and put them together, they do this face touch double than familiar animals, okay? And then they eventually start to fight. What we hypothesize is that perhaps when they're doing this encounter, in the familiar case, they're getting some cue, they're getting some reciprocal signal, so they're like, okay, you're one of me, I know who you are. But with the foreign animal, perhaps they're touching the face and they're not getting that cue that they're used to getting, so they keep touching more and more to confirm, trying to figure out who the other animal is, and when they can't, then maybe aggression is unlocked. That's what we're hypothesizing. But we need to do more experiments to really prove that idea. But what we can say is that this face touch is a prominent part of their social communication. So the last data I'll show you on this is looking at sort of a non-behavioral readout, looking at piezo2, that famous ion channel that was recently awarded the Nobel Prize for its discovery. This is a paper from Artem Patapushian and Kara Marshall, a former postdoc in Artem's lab who has their own lab at Baylor now. What they show in this paper is that there's a dye that is a great proxy for piezo2 activity. So you can see here, so these this dye labels the sensory ganglia and it labels the peripheral tissues. So you can see here, there's a lot of expression of this dye in the sensory ganglia, but when you ablate piezo2, you don't see this dye. And if you give animals touch stimuli, you see this dye really lights up the sensory terminals, but no touch stimuli, it's blank. This dye is called FM143. For reasons that are not fully clear, when the piezo2 ion channel is open, this dye goes into the channel and hangs out in the pocket of this ion channel. And you can image this, and it gives a really quantitative readout of piezo2 activation, mechanosensation. So for us, using a non-model organism, we thought this was a great tool. So we applied this dye in naked mole rats as animals from our colony were either isolated or brought together in a social context. And again, the dye will label sensory ganglia and even the peripheral terminals. And this is what we see. So we take a mole rat that is isolated. So we're looking in the trigeminal ganglion. This is the sensory ganglion for neurons that feed into the face. We see not much activity, right? If an animal is isolated for this is a 24-hour experiment. But look at the mole rat who's been with its friends for 24 hours, right? Quite a striking difference, right? Widespread activity or activation of this dye, which is a proxy for piezo2 activity. Really consistent with this face touch, a lot of face touch occurring in this 24-hour period, and probably using this ion channel piezo2, which is also cool too, that these animals that are separated from mice by 30 million years of evolution appear to be using some of the same molecules. And we can see activation in these sensory endings, these lanceolate endings, which are almost certainly A-beta mechanoreceptive neurons. So an independent readout of the prominence of this social touch. All right, so just to kind of wrap up what I've talked about, with the naked mole rats, we see unique constellations of behavior that stratify by caste. Something else that came out of this that I didn't show at all today, just for the sake of time, was that using their spontaneous behaviors, even in isolation, we can cluster animals according to their dominance rank. We can basically predict their order in that scale based off their spontaneous behavior, which is something that was unexpected for us, which almost suggests that depending upon how dominant or submissive you are, even in isolation, you just have a unique constellation of behaviors, suggesting that it's a whole brain and behavior state that segregates with dominance position. I just talked about the piezo work as well. Some ongoing work we're doing that we're excited about, we've been doing some comparative genomics, and this has been in collaboration with David Ginty and Mike Greenberg's groups at Harvard. And Andrew is a postdoc there. He's been coming back and forth from Boston to Columbia and working with Preston in the lab. And they've been taking sensory ganglia from naked mole rats and doing single-cell RNA-seq and single-cell ATAC-seq to basically get, to see if naked mole rats have the same molecularly defined peripheral sensory neuron cell types as we see in other rodents. And with ATAC-seq, maybe this can teach us about what enhancer areas may be driving gene expression. We want to do, this is a comparative study, but we also want to use this as a way to be able to generate tools. This is a non-model organism, so this will be a heroic effort, but we do want to ultimately do causal-based manipulations in these animals. And we think, having some of these genetic architecture could help us in defining some of these tools. David may kill me if I share some of the early results, but we're here amongst friends. Some of our early results suggest that naked mole rats don't seem to have that gene B4 that I talked about in the earlier part of talking to mouse, and maybe even that entire cell type of mechanoreceptors, the C low threshold mechanoreceptors. And maybe we're starting to think that those CLTMRs are hair specific. So if you have a mouse, a hairy animal, and they have all this fur, you need to evolve a mechanism by which to mediate sensation given all that fur. So then you evolve a cell type that wraps those sensory endings around the hair follicles. But if an animal has no hair like naked mole rats, then maybe you don't need that cell type. That's a prediction, but we need to do more work. But if that's true, then how are naked mole rats sensing social touch if they don't seem to do it using the same classical neurons that other mammals do? That's an exciting question that we hope to dive into. Will has been building, I think this is almost up and running. This is going to be a data nightmare, but to collect data 24/7 continuously. So videography data, all our animals are RFID chips, so we can track where their location is in the colony. Microphones in there, they do have a really exquisite vocalization structure that we want to capture. And we think just taking this sort of large-scale view of what are all the behaviors that they're doing over the course of 24 hours, we think this could be quite useful in discovering new behaviors. And as we get to the point where we can do causal manipulations and even maybe wireless brain recordings, it'll be nice to know, like, what behaviors are we actually studying? So this is a kind of a foundational experiment that we hope will be useful for subsequent studies. All right, so with that, I'll end. I just want to thank the current members of the lab, all shown there. I didn't get a time to talk about everyone's work, but they're all amazing and creative scientists, and it's a great pleasure for me to be able to work with them all. Lab alumni, funding sources, especially Simon's Foundation. There's a junior fellow here, Andre, that maybe some people know, and another junior fellow to come. It hasn't been, I think, officially announced yet. And thank you all very much for your attention.
I want to thank Ishmael for a fascinating talk, and also want to thank his wife for lending him on Valentine's Day. The kids for showing up. I drag my kids to my lectures, and eventually you turn 21, you don't have to come anymore. So we have time for some questions.
Ish, don't they have sparse hairs all over their body?
Yeah, in this, so in this paper, interestingly, they measured this in this 2003 paper. So the morphology of the whiskers, the vibrissae on their face are indistinguishable from the vibrissae on their bodies. Morphologically, they look the same. So in total, they have about 100 of these vibrissae that tile their entire bodies. And like is shown here, each one is exquisitely sensitive. They have a higher collection in their face, but the same sort of whiskers tile their entire body. Unlike other rodents where whiskers are kind of only in the face, I guess their whiskers are like everywhere.
So I just don't see why they don't feel soft touch. They have the hair follicle, right?
Why? So the question is the data that is too new to even report, and Ginty might kill me. it looks like they don't have the CLTMRs, right? But you're saying if they still have those Rebursae, couldn't the same cell type just wrap around that sort of base? Potentially, right? I think what this also gets at is that there must be other sensory neuron types that are mediating social touch. One thing that, and I mentioned that Keystone meeting we were recently at, and there were some people from NIH really pressing us to think as a field, to think beyond these CLTMRs, because social touch is not always like this stroking. Sometimes it's, it's a tighter embrace and it's going deeper, hitting muscles. What are the sensory neurons that are there? So perhaps these animals, the social touch that they use is this not as superficial as it would be in animals with hair. Again, these are early days and it's a little speculative, but we'll see.
I have a quick question. So if two mole rats are similar in ranking, could they disagree on the ranking and fight?
Yeah, so, wonderful question. We don't really see too much fighting with animals in the same colony. Remember, they have to be very agreeable with one another because they never leave the colony. I didn't mention this, but the range of our animals in that colony, I think our youngest animal is 2, our oldest animal is 25. So they're also long-lived. They live like 30 to 40 years. So in one colony. So you live 20 years with an animal, right? So if they fought, this would be a really tough lifestyle. So, I think that test I showed is just a consequence of they're going in those tubes a lot, and the dominant animal is just like, I'm going through faster and I'm going through on top, yeah.
So Ishmael, I was introspecting during your talk, and I don't have a lot of hairy skin, and less than I used to actually.
Yeah.
And humans, of course, experience social touch through hairless skin.
Yep.
And you have the well-documented receptor types and so on and so forth in skin. So this really goes back to Darcy's question.
Exactly.
There must be mechanisms for detecting that kind of social touch in hairless skin.
Again, when David and I were chatting about this result just a few weeks ago, so it's early days, but the first thing I thought, and I was chatting with my lab about, is like, maybe the naked mole rat is a better model for social touch than a mouse, right? This mouse has all this hair and has had to evolve specialized sensory endings to deal with this fur. And we don't have, a lot of hair. And even at this Keystone meeting, there were people are starting to do a lot of sequencing of DRG neurons in humans. And they're seeing that these CLTMRs are not a prominent cell type in humans. Go figure, not as much hair as a mouse, right? So this could be one context where maybe the mouse is not the best model. Mice are great models for lots of things, but if you want to study the neurons for hair, then maybe they're a bit of an outlier. But I don't know. I don't want the field to hate me, but
Aish, thank you so much. That was an amazing talk. Yeah. I have like 100 questions, but I'll ask one very broad question. So pain can be pleasurable in some contexts.
Yes.
Do we know how that works?
That's a great question. The short answer is we do not know. Bella in the lab, who I mentioned briefly, that's largely one of her questions she wants to get at in her thesis. How does the brain separate pain versus pleasure? We talked about it at the Keystone meeting, and I think we don't have the best answers. If you look at some of the brain areas that are important for pain, thalamus, somatosensory cortex, insular cortex, anterior cingulate cortex, amygdala, those same areas are important for pleasure, right? The same areas turn on, but we don't perceive them as the same most of the time. So even in the normal state, how does the brain segregate pain versus pleasure, I think is not worked out. And in those contexts where, as you mentioned, painful things are pleasurable, I think it's anyone's guess. It's a great question, but I don't think anyone has a satisfying, answer.
Over here. Beautiful talk. My question is about the B4 system, and when you were saying the mice that are defective in that, what does their fitness look like? Are they able to mate? Is there a different pathway through which they mate? And if so,
Yeah,
is there something
good
that's
question. We're
compensatory?
starting to look at this a little bit. We haven't looked across, done this systematically across many generations, so it's possible if we looked at 10 generations, you'll start to see litter sizes go down or something like that. So we haven't done it exquisitely, an exquisite analysis, but it appears that there must be some redundancy because although the sexual receptivity is reduced, I guess maybe you didn't see it, but the males don't stop. The dumb male is just like, he's not getting any reciprocation, but he doesn't stop. So they will eventually mate, and the females get pregnant, and they seem to be okay mothers. Okay, when we look at their litter sizes, the weight of the animals, nursing, we can't really find any overt phenotype. Okay,
So there's likely redundancy or compensatory mechanisms?
we would assume so. That's our leading hypothesis. Yeah.
One last question.
Thanks. That was a great talk.
Thanks.
So I have a question about, do you have a hypothesis as to why or how the mole rats are able to determine their social rank? Is it just something limited to touch, or are there other senses involved in that?
Yeah, so our best hypothesis, obviously, I'm a touch guy, so the world's all touch, but it's probably multimodal. So and we are starting to look at this, and there are other researchers who are looking at. We just had Alison Barker here, a colleague in Germany, who co-discovered and extended earlier work showing that they have exquisite vocalization. They have 25 unique vocalizations, right? So it's unclear what those are for, but presumably it maybe have something to do with animal ID. The work on olfaction I mentioned is controversial. There's some papers that show animals do have the ability to use olfaction to discriminate, others don't. In our own hands, we've done like olfactory-based tasks where you take, bedding from their home colony and bedding from a foreign colony. They prefer the bedding from their home colony. That clearly seems like an olfactory-driven behavior. So if I would guess or speculate, I think it's multimodal. When they're coming to each other, they give a vocalization pattern call that they may interpret the other animal as being a member of their colony. Maybe there's something with caste in there. The touch, maybe there's a certain touch pattern, and smell. So maybe these things are kind of all coming together to give some representation about identity. And maybe even in somatosensory cortex, this is speculative, a lot of speculation tonight, but we do speculate with this expanded somatosensory cortex that maybe it's not only detecting tactile cues, but maybe some other like vocalizations or olfaction. Why couldn't they also activate somatosensory cortex? So short answer, but it's a long-winded way to answer it. We think it's multimodal. Touch being prominent, but other sensations as well. Exactly how they ID a foreigner. Right. Yeah. A rich dialect, but they can't really hear. It's something that Allison we were chatting about. It's like, is this, but people say this in the birdsong literature, right? I was talking to our colleague Dimitri at Columbia. Birdsong have this very rich, vocal pattern, but what are they using it for? It's not so clear. The mole rats don't seem to hear so well, so what's the use of having an exquisite dialect? But there's a lot left to be discovered, I would say.
So thank you for a stimulating talk. I mean, and really for sharing lots of up-to-date, like hot off the press data, which is always really fun to hear.
Hopefully it stands the test of time.
And I guess one can't help but say it was touching. Thank you.